Thanks, Marta, for a very gracious introduction. It's a little bit terrifying to do this, but I really appreciate the invitation. And again, thanks to Marta and Michael and Sylvain and Lena for all you've done to bring us here. We're really enjoying the meeting, and it's a real pleasure. Appreciate it. So for the next 45 minutes, and I promise not to go over, I'd like to echo what Dr. Elvisha Damala said about diversity bringing us together. I mean, this has been a real thrill to be part of a worldwide collaboration in the global quest to map human brain diseases. And I'll tell you a little bit about the ENIGMA Consortium. So for the last 15 years, ENIGMA has published large international neuroimaging studies of over 30 brain disorders. Many of you have contributed, led, taken part, done the hard work for these, analyzing 200,000 people's brain scans from a little over 1,000 medical centers. And this has led to the largest MRI studies in neurology, Parkinson's, epilepsy, ataxia, brain trauma and stroke, in psychiatry, schizophrenia, bipolar major depression, PTSD, and addiction, and also neurodevelopmental and neurogenetic disorders. So the overall goal has been to say, how do these 33 disorders affect the brain, at least as seen with MRI, diffusion tensor imaging, resting state, MEG and EEG? And then another challenge is, how does genetic variation affect the brain by screening a lot of the variants in our genetic code, the copy number variants, the deletions, the common variants, with the goal of finding genetic markers and environmental factors that affect brain development and aging, disease risk and progression and treatment response. So now ENIGMA isn't the solution to everything. There's plenty of good studies that are done with a more focused design, but it is good for enhancing statistical power, efficiency, team science, the collective expertise. It's not just the data, it's the points of view and the diverse points of view. So I'll tell you about ENIGMA, how the neuroscientists around the world work together, tell you a little bit about how the results on different diseases interrelate. We'll talk about how genetics is used to analyze brain images, and then some challenges, but also some advantages of pooling data internationally. And we'll wrap up with something on AI and how that might be able to help us. So here's where the data is. So there's not a spot where people have centralized this data. And all over the world, people are collecting brain scans all day, every day from different clinical populations. The color here is the disease that each center specializes in. And this has been written up as a sort of logistic feat to try and integrate data that is not centralized. Many of the reviews of how ENIGMA works have talked about crowdsourcing, getting data into the hands of many people. And of course, OHBM has led the way in opening up science and making sure people can have access to data. And also with crowdsourcing comes a responsibility. So people with good ideas can come to the table, bring their insights and bring it to bear on data that they didn't have to collect themselves. So if you're interested how this works, it's a rather complicated paper on how this data came together. And big thanks to Gary Egan, who had the idea of having a special issue of human brain mapping. I would say that a lot of these papers take about, I won't say 7 years to complete, the slowest one was 7 years to complete, but because there's a lot of sites, a lot of people involved, it does take a while. So ENIGMA has 33 clinical working groups, and you can see in the middle the disorders from neurology, things like stroke, epilepsy, Parkinson's, frontotemporal dementia. And around the perimeter, disorders such as schizophrenia, bipolar, depression, things where there isn't an agreed or established brain imaging biomarker of those disorders. And some of these groups have subgroups. So if you have a brain injury, you might have incurred it in the military, or it's very, very sad, intimate partner violence, domestic violence, occasionally leads to brain injuries. And also on the sports field, you might be a sports participant who has a concussion. So we're not behaving as though these disorders are homogeneous. There's many different etiologies. And this is supported by 18 technical working groups. So Steve Smith was just awarded the Glass Brain Award. I mean, we rest on the shoulders of giants here. I mean, there's so many people creating software that we all use every day, and so many of these things are made possible by the efforts of many of you. The technical group supports standardized processing and QC and harmonization of all these different methods. And you can see some of the leaders in this activity shown here. I would say that these projects are led from all over the world. There's some people here in different parts of the world. This one is in the Middle East. There's meetings at OHBM that are very common, where people can get to meet each other. And ENIGMA is collecting data as well. So ENIGMA India is collecting brain imaging data in India. Mohammed Parvaz, who's here, is leading with Maheen Adamson a new data collection project in Pakistan. And Emily Dennis has launched an outreach effort called ENIGMA U that teaches high schoolers and undergrads just getting into neuroimaging the basics of the field. She's done an absolutely terrific job. This is a free course for anyone all over the world. And if you have undergrads that want to brush up the brain imaging, go and have a look. So we usually meet at OHBM. If you're allowed to bring a drone into the country, Andrea Rimia, who's here somewhere, will make a video. And the topology of scientific collaboration is quite interesting. So if you have people who are expert in structural MRI, DTI, resting state, that study a particular disease, they might want to compare their observations across diseases. But you still need to harmonize the analysis workflows. The results have to be comparable from one study to another. And so let's begin to look at the different results. So this is ENIGMA studies of 9 different brain disorders: major depression, bipolar disorder, schizophrenia. The colors are going to be the cortical thickness deficit in each of these disorders. And everyone ran free SOFA. They've done this in a meta-analytic framework to see what is the first look of what these diseases look like. And you can have in fact, there are t-shirts. I think if you want a t-shirt, you can get one on the way out. So this is the new ENIGMA t-shirt. So there are, thanks to Neda Jahanshad, who's coordinated ENIGMA DTI for over a decade. And again, thanks, Steve, to your team developing TBSS and FSL. There are now global DTI studies of 18 disorders with over 80,000 people. And the ability to do any of this goes back to Jean Talairach. So this lecture is named after a prominent neurosurgeon and, in fact, a psychiatrist, Jean Talairach, who was a member of the French resistance during World War II. If you want to learn about him, read Bernard Mazoyer's retrospective on his life. But when I was a grad student, there would be a Talairach atlas on the desk. And Talairach observed when he was doing deep brain neurosurgery that if you could overlay the anterior commissure and the posterior commissure of that patient's pneumoencephalogram onto all the aggregated drawings, diagrams, histology, architectural maps, this would be a reference system that could become the basis for collecting observations both neurosurgically and neuroscientifically. And this was then built on by Ellen Evans, who's here. Actually, Ellen Evans is here, who with Arthur Toga and Peter Fox and many colleagues developed the ICBM project in the 1990s, where this was very, very exciting. You could take a digital template, and through mutual information-based registration, you could line up your PET data, your SPECT data, your MRI data with that template. And then with Carl Friston's team and the people in London developing SPM, Steve's team developing FSL, you could create statistical parametric maps, not just with in vivo imaging. So Katrin Arments, Alan Evans and their team, Katrin's team in Jülich, has been working on big brain, layering on 20-micron histology, receptor maps, chemoarchitectural maps, all of the aggregated neuroscientific mapping that you could bring into this space. So let's go back to Dr. Talairach and what he proposed. So if you or I were scanned today, there'd be a principled way of lining up that scan into a common reference. And you could do group difference analysis. All of you know this. I don't need to teach you it. This is a study of Alzheimer's disease cortical thinning. And in Brisbane, in the late 90s, here at the Queensland Institute of Medical Research, Greg Duzubikare, who may be here, started scanning Alzheimer's patients longitudinally. And he was able to reconstruct with his team a time-lapse film of the progression of Alzheimer's disease in the brain. Judy Rappaport and the team at NIMH scanned children with schizophrenia, and she was able to reconstruct with Jay Geed, also with the McGill group and many others, the trajectory of the spread of schizophrenia. And drug trialists began to use these. So Lilly was testing a novel antipsychotic, and they were able to ask in this statistical reference space that was pioneered by Talairach, where is it that the novel drug olanzapine slows the spread of gray matter loss? And these types of maps, compiled from many, many studies and many, many sites, perform the basis for statistical mapping of brain disease. Now, the problem is, you all know that aging produces brain changes as well. So this time-lapse movie of normal brain changes has to be built in. So there's a lot of interest nowadays in normative modeling. So Richard Bethlehem with the work on brain charts in Nature, André Marquand in Nijmegen with his work on the PCN toolkit And Sophia Franco and Rui Yang, who work on this in ENIGMA, have compiled these reference charts. These are healthy people. There's data from around the world. You can have centiles. You can norm people's, in this case, structural imaging, but you do this with other types of imaging. You could look at the trajectory throughout life for different structures and how they develop. And you could essentially look at the recovery, remission, or deterioration of individuals within this statistical reference system. And if everyone was the same and all the sites collected the same data with the same distribution, that would be it. But a great deal of work has gone into statistical harmonization. Taki Shinohara is here, Johanna Bayer is here, Philip Simon is here. They have written extensively on how you integrate multi-site data into a common statistical model. And it's not that simple. So if you or I were scanned in Los Angeles or in Brisbane or somewhere else, the population and the scanner and the protocol might lead to systematic differences. So over the course of time, combat, normative modeling, even AI has been brought to bear on this. And Neda Jahanshad, who just won the Open Science Award, has led the way here with her student Alyssa Zhu in taking all of the piecemeal data from the lifespan, from the different sites, and putting it into a common model that then can act as a reference for a study. Many of you will know about André Marquand and his team in Nijmegen and their normative, their hierarchical Bayesian model, where you don't just have a reference, but you can adapt it to the data that you're collecting as a normative model. And this can be used to assess abnormalities in new subjects. So ENIGMA doesn't just do structural imaging. There's just an example. There's MRI, DTI, resting state, MEG, EEG. What do we do with our scan? So if you want to join in ENIGMA, if you have a good idea, you can run free surfer, fMRI prep. You can test associations between brain measures and things that you care about, clinical factors of interest, risk factors, maybe in treatment. And the first and simplest type of analysis is a meta-analysis. It's a weighted vote. So if you run VBM on data from, how many sites is this? 37. This is work by Matt Kempton's team. The p-values get better and better. I mean, they just become astronomically good after a while. And this is a VBM study of gray matter anomalies in bipolar and OCD. If you're to do this across the world, nobody presupposes that all the patients are the same at every site. So let's take an example. This is 33 sites in ENIGMA schizophrenia. They all run free surfer, they QC it. And 25 of those 33 sites individually found a statistical difference between schizophrenia and healthy people in terms of the regional cortical thickness or volume of those areas. The whole brain is affected. If you were to do this in different disorders, and we'll go through them, schizophrenia, OCD, neurogenetic syndromes, epilepsy, if you were to just average the data together and meta-analyze the statistical effects, you would see a mosaic of differences. The disorder with the most striking effect on the brain, phenomenally severe anorexia, has in some cases an optimistic story. The brain almost fully recovers after 3 months of renutrition in this work by Stefan Ehrlich and Esther Walton. Another general principle is that diseases with larger effects on the brain tend to be less common. And so if you were to look at the effect size of the largest brain difference on MRI so far detected, and the prevalence of the disorder on the other axis, you'd find neurogenetic syndromes, you'd get slightly more prevalent psychiatric syndromes, very, very prevalent disorders such as anxiety, such as I'm going through now, are common, very common, and they have lesser effects. And so Leanne Schmal in Melbourne set up the ENIGMA Major Depression Group. They have found profound differences, very systematic in the cingulate, the limbic system. Chris Ching, who leads the bipolar group, finds frontal differences. And if you go into tractometry, you see that the microstructural differences in bipolar in the frontal lobe are more profound than in depression, which makes sense given the disinhibition in mania. And you can then parse deeper and deeper. You would say, go and find a track, go and find a microstructural signature of these disorders, all the while drawing on all the mathematical innovation that has gone into harmonizing data, in this case, point by point along tracks, to make sure that you can model all of the influential factors. So going back to that principle, the more common the disease, the less the effect is on the brain, which is a general principle. Schizophrenia's effect tended to be about double the effect of bipolar disorder, which tended to be about double the effect of major depression, with ADHD being very, very different. But this meta-analysis is a bit troubling. So Wei Cheng and Yu Chao Jiang at Fudan University in Shanghai said, you know what? There are subtypes. We looked at 41 cohort schizophrenia data. We're still looking at FreeSurfer, but we've run a staging and subtyping effort. They also included a large discovery cohort from China that they had been involved with collecting themselves. And they were arguing that there are two subtypes of schizophrenia. In the first, which is more common, the trajectory of gray matter loss originating in Broca's area and then proceeding posteriorly into the rest of the cortex. And in this first one, there was a greater prevalence of anxiety, depression, negative symptoms, whereas in the other, which originated in the temporal lobe, there seemed to be a more mild trajectory. And this was found in China, and it was found in the rest of the world. No trouble replicating it. Again, depression and anxiety only worsening in type 1. This was replicated. The differences across cultures seem to be fairly minimal. Now, contrast this with something like Parkinson's. So in Parkinson's disease, the stages are defined clinically. And so ENIGMA PD has been looking at the trajectory of cortical atrophy from Hoehn-Yahr stage 1 to stage 5. Atrophy looks like the BRAC maps. Atrophy of the basal ganglia is spreading from the caudate putamen to the rest of the subcortex. The cerebellum has this traveling wave of atrophy, which was very surprising when first seen. And again, the white matter microstructure has this traveling wave in Parkinson's disease of progressive deterioration. Now, Alan Dagger at McGill said, do people at high genetic risk for Parkinson's have any of this? And he was able to show that people at high polygenic risk for Parkinson's in their teens already had a subsignature of PD. It was very large overlap, at least in terms of its topography. Boris Bernhardt, also at McGill, looked at polygenic risk for epilepsy, and he and his team were also able to find that people who were healthy but had very high polygenic risk for epilepsy had a subsignature of the disease with very, very high spatial correlation to the illness. Sophie Volk, who was also here working with Hugh Garavan, she was looking at people who are addicted to alcohol, cocaine, cannabis, opiates, nicotine on a scale never before seen. Hugh Garavan has been aggregating this data with Anthony Giuliano at University of Vermont. Look at how all of these disorders are associated with frontal cortical alterations. Is it the substance? Is it disinhibition? It's very interesting. If you are looking at other disorders of disinhibition, there's conduct disorder, antisocial personality disorder, there's a number of externalizing disorders which have to do with the symptomatology often emerging in adolescence, that in the brain maps is associated with a surface area reduction that preferentially is found in the frontal cortex, very similar to the addictive disorders. And again, many of these cross-disorder studies are being led to find these signatures. The childhood onset disorders tend to influence surface area more than thickness. This is Marlena Stagidis and Yidian Gao from Birmingham University in the United Kingdom. They find that in a lot of the externalizing disorders, there's a deficit in cortical surface area, and they've tracked this across many, many disorders. Seb Jacquemont, Sarah Larivière, Kuldeep Kumar at University of Montreal have been asking what are the general principles. In disorders of early onset, there tends to be a surface area deficit. In disorders of adult onset, there tends to be a thickness deficit. And if you were to try to put new disorders within this framework, Haley Wang and Tiffany Ho, who are here, they were interested in childhood sexual abuse, childhood neglect. And within this picture, the deficits primarily are surface area deficits. They occur early. They occur before adolescence. And again, if you were to try to systematize these general pictures coming out through ENIGMA of all the disorders, very early prenatal onset tends to be associated with unimodal, transmodal, universal surface area deficit. Childhood onset, lower surface area. Adult onset, alterations in cortical thickness. So many of you attended the multimodal workshop today. So the gradient people came to that, and they are expert in trying to see patterns in the different disorders. So Sophie Volk, Boris Bernhardt, and many of the advocates of gradient analysis and brain mapping, originally based on work by Dan Margulies, who many of you know, have tried to ask, do disorders have a predilection for unimodal cortex, association cortex? Does it depend on the timing? And they've decomposed these disorders in ENIGMA into 3 signals: a general one that is found in every disorder, like the P factor, one that's specific to the disease, and one that medication is answerable for. And I strongly encourage you to learn about this. ENIGMA is just beginning in this area, looking at gradients, but this cast of characters, truly brilliant folks here, again, have this holistic way of looking into the alterations. One of the things that seems to appear from this is that the cortical organization follows continuous gradients from primary cortex to heteromodal cortex. And when the disorder strikes, it's often interfering with the developmental process that is situated in this gradient hierarchy. Bratislav Misek and Justine Hansen have been relating this vulnerability pattern to receptors, to chemoarchitecture, to myelin architecture, and they also have a different take on this. They say, well, for cortex to be vulnerable, there's 2 things that might be occurring. There might be a local enrichment of receptors that are important for that disease. It might be GABA, serotonin, glutamate, dopamine that might predict vulnerability to different disorders. And to a lesser extent, at least in psychiatry, more so in neurology, the network connectivity of a hub is influential in determining how vulnerable it might be to disease. So if you want to use these tools, Sarah Larivière, huge credit to her, also with Bratislav Misek and his NeuroMaps toolbox. If you've been making brain maps from in vivo data, you can go and look at all of the aggregated molecular, chemo, architectural, and receptor data and bring them in to correlation with your own studies. What affects disease? So ENIGMA codes medication by mode of action. So there's a lot of medications. In fact, I just took some Ativan. So Ativan is a beta blocker, but it has other receptors it binds with. So one of the things that has been done in ENIGMA is to try to stratify people on different medications: antipsychotics, neuroleptics, anticonvulsants, antidepressants, lithium, by mode of action, and overlay the receptor targets of these. Thomas Hayek and L'Oreal Dietz have said that a large part of the deficits we see in psychiatric illness are moderated or mediated by lifestyle, body mass index, cardiovascular health. Many of these somatic factors causally worsen the deficit that you see in the brain. Access to healthcare. Heath Pardo, who's here. Now, this is a rather eye-opening result. When he was looking at the effect of epilepsy on the brain all over the world, he had the idea to link the degree of abnormality with the income of the country. And he found that perhaps due to access to healthcare, the deficits were more profound in countries with greater income inequality. It makes it very, very hard to dismiss that as a factor. Very, very important to model social and environment determinants of health. Age of onset. Someone who's been ill a long time, their disorder might interact with aging. And what was almost universally found in ENIGMA is that people who'd been ill a long time had more severe deficits. You don't want to meta-analyze that. You want to understand the effect of age. And Ian Harding, who was at Monash when he did this study, he's now in Brisbane. He has been looking at the ataxias. Ataxias, many of them come from neurogenetic alterations, but the duration of illness, the age of onset and the duration of illness have a profound effect here in the cerebellum of where those effects might be. Severity of injury. So some disorders, not all these disorders are alike in their etiology. Some disorders, such as traumatic brain injury, have a precipitating event. The severity of injury is of great interest to those of you who've had a concussion. So Emily Dennis and her team, this paper led by Finian Kelleher, found that people with mild traumatic brain injury, this is a study of children, tend to recover. And when they were scanned longitudinally, in this case the cerebellum showed a profile of remodeling and regeneration, whereas those with more severe traumatic injuries tended to degenerate from the acute to chronic phase. Genetics. So this is what got ENIGMA started. So you could basically look at every SNP in the genome, every common variant in the genome, and do an SPM with it. You would say, does this genetic variant affect the brain? Let's run this 3 million times. Nick Martin, who is a very brilliant Australian geneticist and was the president of the Behavioral Genetics Association in 2009, said, well, why don't brain images do genetics? You should do genetics. And in fact, many of you do genetics now. But he did say, If you want to discover genes that affect the brain, you'll need brain images from over 50,000 people. Just because images are more expensive to collect than measures of height or weight, it doesn't change the power calculations. Ouch, ouch, imaging is expensive. So in Nature, this is a study with 287 authors, wow, that goes along the genome and asks the following question: does the nucleotide present at this point in the genome and its common variation affect the volume of the putamen, the caudate, the thalamus, many of the structures that we care about? And what you do is this: it's like SPM. You go along the genome, and all of the variants that we share are not variable, but the ones where we do have variants, they can be associated with the structure of the brain in this case. This paper in Science, which took 7 years, sorry about that, everyone who had to put up with it, was led from USC by Neda Jahanshad and Katrina Grasby from here in Brisbane. In fact, here are some of the co-authors. They found that if you were to isolate the genomic loci that were associated with volumetric differences in the brain, over a third of the variation in cortical surface area was explainable just from genetic variation. Around a quarter of the variation in thickness was explainable just from common genetic variation. And there were hotspots that affect many, many cortical regions. And if you were to travel around the cortex and say, tell me the genomic loci that are associated with this one and this one and this one, there was both common and distinct genetic architecture such that you could cluster it. So you could say, if the genetic commonality between different cortical regions were clustered, hierarchically aggregated, it would have a modular architecture. And this work went on in the hands of Anders Dale's team at UC San Diego to argue that there are genetic programs that influence sectors of the cortex, and they may or may not coincide with the atlas definitions. These are the loci affecting subcortical volume, a recent paper in Nature Genetics by Luis Garcia Marin. You can run SPM on these, so you can say, here is a polygenic risk score that we've discovered through running FreeSurfer. Let's go map it back into the brain. And there seems to be exceptionally segregated genetic programs. So hippocampal SNPs affect the hippocampus, the putamen, and you can predict up to 10% of the variance in these volumes from a polygenic score. Even the changes in the brain, people scanned twice in a study led by Rachel Brouwer in the Netherlands. In 40 cohorts of people who were scanned twice at an interval of 2 years, she discovered 15 genetic loci, one of them ApoE, which is a haplotype which was very famous for affecting your rate of brain atrophy in old age, but another 14 that are common variants that affect the speed of brain development in terms of volumetric growth. They went on to look at EEG, so looking at resting state EEG. The most common variants that influence EEG are profoundly implicated in epilepsy. So the inhibitory neurotransmitters that regulate the propagation of seizures in the living brain. There were many variants in the GABA2 receptor subunit. So this is something that is implicated in faulty inhibitory neurotransmission, strongly implicated in epilepsy. Polymorphic variation at this gene alters the power spectrum of EEG profoundly, even in healthy people. Now, if you go to Iceland, you might hear about this gentleman. Kari Stephenson runs Decode Genetics. So once the genetic data had been amassed from a large number of sites, he and Ida Sonderby, who is also here tonight, were interested in whether deletions of the genome, these are rare events in the genome, were associated with the same profile of deficits in the brain as the disorders that those deletions put you at risk for. So Kuldeep Kumar and Claudio Maldonado made a library. So they ranked all the genetic deletions, none of them are very common, in order of prevalence, and they clustered the patterns in the brain and found that the idiopathic disorders that these deletions put you at risk for, 22q11 deletion syndrome for schizophrenia, many of these have a high odds ratio of risk for neurodevelopmental or psychiatric illnesses. There was a fundamental genetic clustering among the deletions that mapped onto the disorders that idiopathically arise for non-genetic reasons. And this whole program of activity, again, is immensely exciting and only possible when people put their data together. Anna Silva and David Linden and ENIGMA CNV were interested in where copy number variants in the genome have the greatest effect on the cortex. And secondly, whether or not there is a region where copy number variants influence it that have the most profound and highest odds ratio for disease. And they found that almost in a linear way, the copy number variants that affect occipital cortex, which of course has very profound functional effects, tended to greatly increase risk, often 10, 20-fold for schizophrenia. And this is a hugely interesting program. Methylation of the genome. So you're very welcome to look into the literature on epigenetics, the epigenetic clocks. Sylvain de Rivière in King's College London, Di Chen, who I talked to earlier today, have found strong links between brain morphometry and the methylation of Hox genes, homeobox genes. If you go back to your developmental neuroscience class, Hox genes involved in regulating the segmentation of the mammalian brain. And they've even been doing VBM. You need maybe 3,000 people to do this. All right, so that was all massive crowdsourcing, a lot of people analyzing a lot of data in a lot of countries. How can AI help with all this? Now, I don't begin to say I'm an expert in this, but it's exciting. I find this very exciting. In order of difficulty, I think AI could help us with 3 problems. One is with diagnosis and prognosis, and many of you are presenting at this meeting these beautiful algorithms using deep learning that identify features that none of us would ever have in mind, but end up being profoundly helpful for diagnosing at least neurological disorders, perhaps more challenging in psychiatry. In imaging genetics, could we use AI to discover genomic loci? And then, of course, an area that I don't know anything about, but many of you do, brain decoding, understanding the fundamental latent representations in the brain and their relationship to either perception or action or things that are in the external world. In AI, you often take all of the aggregated biomarker data from brain imaging, genomics, omics, clinical, even actigraphy, and you'd like to predict the diagnosis of the patient, their prognosis, their treatment response, either because you're a physician and you want to make the decision, or because you're a scientist and you want to find either mathematical techniques to recover this, or data you should collect, biomarkers you should collect to diagnose these things. And so in this film was made by Arthur Toga in Los Angeles. This is how these deep learning methods work. You would have this gigantic mathematical function, maybe with 1 million or 10 million parameters. You'd feed in brain images on one side. You would try to calculate a mathematical function that will map that image onto a decision. The decision might be Alzheimer's or not, or it might be likely to recover or might not. Xiaogan Yan, who is here today, this is, I think, the 3rd largest brain imaging study ever published. He trained a deep learning algorithm on MRI data from 217 sites, and with 91% accuracy was able to diagnose Alzheimer's disease in patients from a sight and scanner that algorithm had never seen. And then with explainable AI, which is a very large field, they were able to occlude parts of the image, and the parts of the image that made the most difference to the classifier's decision are highlightable as a salience map, an attention map, something that will give you a guide as to what features might be valuable here. AI that reads brain scans shows promise for finding Alzheimer's genes, so what's going on there? So Nature News said, what about you do this? You could write a classifier, a deep learning classifier or a machine learning classifier, that can detect disease in MRI scans, and then you feed its opinion into all of the genomic screens, the environmental screens, the epigenetic screens that we've been talking about earlier on. And this was first done by Andy Sakin in Indiana. Many of you know him, he's a prominent Alzheimer's expert. He sought with deep learning the tau PET signature that was most strongly associated with Alzheimer's, and then gave that a score and fed the tau PET score into a CNN that searched the genome for markers that were influential in promoting amyloid deposition. And Tai-Ho Zhou, who's a prominent AI expert, is leading this project. You could imagine this would be applicable to any type of signal in brain images and any risk factor. Frightening new AI can recreate your face by analyzing your DNA. That sounds awful. So a diffusion model that is called DIFFACE was trained on a little short of 10,000 genome face pairs, mapping genomes to the 3D scans. And the relationship between the genomic variation and the phenotypic variation was discovered by contrastive learning. So what's going on here? So let's try and understand what's going on here. So this is a person playing the piano. But you could imagine a chord, if you were to go to the genome and find all of the loci that influence a particular part of the brain in proportion to the degree that they influence that part of the brain, there would be a polygenic score, or a chord, in the genome that would maximally associate with, in this case, it's a VBM signal, it could be a functional signal, it could be an amyloid signal in a particular part of the brain. So cross-modal transformers, which are a variation on the standard language transformer used in ChatGPT, can be used to learn gene-brain associations. There's a linear projection of the genome. We make polygenic risk scores, which Alan Dagger and Boris Bernhardt said already burn a signature into the brain prior to the illness coming on. So these linear projections of the genome are used. Why couldn't you search the set of all linear projections of the genome and the set of all linear projections of the image and find the best one, the one in which there was the greatest correlation? And you can do that. So cross-modal transformers can learn associations between genomic variation, queries, imaging variation, keys, and transformers compute learn projections or embeddings. So whenever these deep learning methods apply, even to language, they take all of the syntactic structure of your vocabulary, map it into a high-dimensional embedding space, and they seek links between that embedding and another representation which has been simplified. Earlier today, in the modal imaging workshop, Vince Calhoun talked about this. Vince Calhoun developed GIFT. GIFT will compute ICA components for you. It's beautiful technology. You can, given two modalities, produce a projection of each that then will find associations between each of these different seemingly disparate modalities, either if you do genetics or if you do brain mapping. So this gentleman used to ride the elevator up to my lab every day, and I'd say, how's your work doing? And he invented the transformer, not when he was at USC. Ashish Vaswani invented the transformer, I think, at Google. And a question that arises if you use these transformers to embed one data modality and relate it to some other data modality is, what are the sample complexity requirements? What are the data requirements? I mean, Google can train on the whole internet. Do we have enough data? Now, the linear case already works. So we found that either CCA, if you work with Vince Calhoun, or if you're doing ICA, there's joint clustering of genetics and imaging that is already able to lock onto a signature that bridges the two modalities. The bioinformaticists say, well, you could make this easier if you use bioinformatics priors. The deep learning people said, well, what about parameter-efficient fine-tuning, which is a way to train these models on very, very limited data? What about generative AI? You know that you can take a painting and turn it into a photograph. You can take a photograph and turn it into a Monet painting. This general idea of style transfer, neural style transfer, capitalized on the ability of convolutional neural networks to distill not just the content, but also the style, the high-order context in the images. And this has been used to make synthetic PET data from MRI. So if you were told that you could synthesize an amyloid PET scan from multimodal MRI, you'd say that is not possible. MRI is not sensitive to amyloid. But by training paired data sets, and you can find out whether you did a good job, you can take the PET scan from someone who did have one, you can come very, very close. Again, this is a cross-modal transformer. There are diffusion transformers that were used in this case, but there are other types of transformers that use projections and look at the mutual information, going back to the 90s when Alan Evans was using mutual information to align brains with the Talairach template. The mutual information is something that these embeddings lock onto and build bridges between these essentially disparate modalities of imaging, but allowing them to be interconvertible and synthesized when it is otherwise too difficult. What mental state is a person in? I would not pretend to be a functional imager. All of you in this room know functional imaging more than I do, but Xiaogan Yan and his team in Beijing trained a transformer to predict the next frames in resting state fMRI. Now you say, why would you do that? Language transformers, when they are using your prompt in a chatbot, are trained by occluding tokens, usually words, in a sentence. And by understanding the fundamental logic of these time series, Xiaogan Yan was able to pre-train a multimodal image transformer to infer what mental state the person was in. Now, in this case, he was studying depression, so he used self-report data. Is the person happy or ruminating or anxious or dissociated or recalling memories? Now, many of you are taking this into whole new landscapes of discovery. The stimulus itself, the content of visual memory itself. But this is immensely useful in psychiatry. I mean, imagine using this as a vector readout of someone's fMRI scan, if with a fair degree of confidence you could infer the mood state, or even the fraction of the time the person spent in that mental state. So just to wrap up, I think as Elvisha Damala said, it was a very inspiring thing that she said. I mean, the diversity of perspective is more important than the data. The data is very important, but you could hoard data. And it'd be a little bit like saying, I want to become good at chess. I've bought all the chess pieces in the world and I'm hoarding them. And that wouldn't do. You need to play chess with other people. So global collaboration, and maybe AI, if you're enthusiastic about it, can now learn from more data than any single lab could ever collect on its own. And it's not just the data, it's our collaborators. Many of you who share this vision and have made these collaborative projects possible bring thousands of years of collective expertise. Just in this room, there are thousands of years of collective expertise. It's incredible. Diverse perspectives. You may have had educational experiences that are unique to you, that would give you new insights on how to proceed. And in difficult times politically, science is a powerful bridge, as you all know, between cultures. Obviously, I'd like to thank Michael and Lena and Sylvain and Marta for bringing us together. You know that this powerful bridge is a bridge that remains open when the stormiest weather is going on in the world. You can connect with people here. We will help to introduce you. I mean, come up to any of us and we'll introduce you to people that you might enjoy working with. And we can support each other. And I think in a political climate that is quite divisive, the community that we have here is really extremely special. So individually, we contribute little to the quest for truth, but working together, the whole vast world of science is within our reach. When do you think that quote was written? It was written a very long time ago. And it's written in Aristotle's Metaphysics. So thank you to ENIGMA, all of you who've made this possible. It's been a remarkable journey. To all of the public and private funders worldwide, to Arthur Toga for directing our institute, to Neda Jahanshad for being Associate Director for ENIGMA, Sophia Thomopoulos for being our project manager and air traffic controller for ENIGMA, and all the research participants worldwide who gave their time and support to these global studies. And if you'd like to come to the ENIGMA social event, it's tomorrow, 7:15, Brisbane Brewery. Don't all come at once, but it'd be lovely to see you. Thanks very much.
Fantastic talk. Thank you so much. So we're going to use the app to have the questions rather than the microphone. So some of you have already started putting those questions in, so keep going. And we've been told that we have a lot of time for questions, so that's bonus.
Thank you very much indeed, Paul, for this very inspiring talk and initiative and adventure journey with ENIGMA. So like Marta said, please post your questions using the app, and maybe we could start with a question that is related to basically the profusion of results that ENIGMA has enabled. And you've shown us, I don't know how many thousands of images today in your presentation, mentioning, very exciting, very novel results just by leveraging big data. If you were to pick one image or one result, basically from 10 years, 12 years of ENIGMA, what would be this result tonight?
Thanks. Well, that's such a nice question. And the one discovery that we didn't think possible, but it was exciting, was the early discoveries of genomic association with the brain. And I think many of you have experienced a similar thrill of discovery where something that you maybe don't think is going to work. And many of you have alluded to the replication crisis. So certainly in neuroscience and other fields, there's been trouble sometimes, at least in the early days of replicating results. So genetic associations were famous. There would be results where the serotonin transporter polymorphism is linked with the size of the hippocampus, or not. And then there'd be results where other polymorphisms in famous candidate genes were linked with imaging. So that whole field became a little bit better organized, much as we do when we get together and analyze data together, to produce really the first replicated genetic associations. Now you could say, well, that's not a big deal. I mean, the effects are very small, but it's the beginning, it's the seed of a project where you could then go into that data and look at methylation, genetic deletions, other types of observations. So I think in summary, if you find an effect that is so small that nobody ever believed that you could pick it up, that was actually, in fairness, what got ENIGMA started. I think people's interest in doing rigorous reproducible studies using genomics began to initiate the incentive for people to get together in a global quest, essentially to look for genomic effects on the brain. Thank you.
So the next one is about cultural differences in diagnosis. So different countries and cultures might have different diagnosis criteria. And so the question is, can you comment on how this might affect the findings that you see across the different psychiatric diseases?
Yeah, what a great question. So Marta's question, thank you for the person asking it, was whether diagnostic differences across the world, I mean, are they important to model? I think the first order, we were very surprised that when Xiao Ganyan's study took place in China across many sites, this is a study of schizophrenia, all of the aggregated data from the rest of the world was revealing essentially the same signature to first order. So at least the reassuring thing in that context is that the basic effect of the disease looks very, very similar, at least in terms of brain mapping. Now, that is not the case in genetics. So you're all well aware that the ApoE4 risk gene for Alzheimer's disease has profound effect if you have Asian ancestry, medium effect if you're of European ancestry, and moderately low effect if you're of African descent. So in genetics, this had been bothersome that there might be ancestry-specific or ancestry-modulated effects. So at least on the brain mapping to first order, this is showing consistent effects. One thing that was a stark reminder that that isn't the full story is Heath Pardo's work. So Heath Pardo was looking at epilepsy. And in the countries where people had less access to healthcare, the effects were absolutely profound. And this would completely be missed by meta-analysis. If you were just asking, can you tell me overall what is going on? And I think that's a salutary reminder. We tend to think that people that are in parts of the world that have the same physicians and same diagnostic criteria have the same illness. But these factors are tremendously important to look at. The third strand of this is medication. So the preferences in different countries for different treatments, particularly in psychiatry, is somewhat different. And so I just want to shout out a word of thanks to Chris Ching. He has been overseeing the aggregation of medication data across the world. And he pointed this out. He says the way people are treated with depression, whether they have access to TMS, whether they have access to even to medication, is different. HIV was one of the disorders we studied. Neda Jahanshad led the ENIGMA studies on HIV. In certain parts of the world, it is taboo to take antiretroviral medicine because of the taboo associated with HIV. So I think that's rather a tantalizing question, because if you didn't think about it, you wouldn't go after you wouldn't go after factors that might be important for modulating the things that you see in the brain. But in the end, they end up being more influential, perhaps, than the genome or other factors that are easier to analyze.
So the other question, the next question, Paul, is not a question, it's a request. We are getting about 100 requests from the room to get a copy of your slides. Is that something possible? But the true next question is concerns, how you foresee bridging between the massive association studies that the ENIGMA Consortium has run over the years with the prospect of applications, especially in the context of personalized medicine.
That's a great question, because it's one thing, I think, Sylvain, as you've said, to discover markers that are associated with a trait. If the markers have a bearing on a patient's response to treatment, they're immensely important. So for example, in ENIGMA, when there are markers that influence the speed of brain degeneration, the first thing that could be done there, and this is a large priority for pharmaceutical companies, is to enroll or make sure you're enrolling people that are exceptionally high risk for the disease progressing. And many of you are familiar with this in the context of dementia. People at very high genetic risk through ApoE are selectively over-enrolled in drug trials. If the whole landscape of the genome were scoured, and you could find additional markers either in a polygenic score or additional plasma markers that might be indicative of someone's likely progression, this would enrich and empower many of the studies that are going on today, and that is even without perfect assessment of these biomarkers. The other one that I think is a little bit more intriguing is even trying to convert some of the genomic signatures into a brain risk signature has been immensely difficult. And all of you are modelers. I mean, you think about things in novel ways. The first series of experiments has been mass univariate, looking at this SNP, looking at this voxel. And this is because we're very reductionistic. But many of you have seen ways that are holistic and integrative and bring many branches of neuroscience together. And it could be, certainly in the maps that we showed today, that there are certain functional systems, they could be gradients, they could be gene expression patterns. There are many, many ways of looking at these multimodal characteristics. And simply by human insight and study and breadth of knowledge, you might be able to see subtypes in the data that might have a differential treatment response. And there is a proof of that in the field of dementia. I mean, we don't think of dementia as a unified entity. We think of it as Alzheimer's, vascular, frontotemporal dementia, Parkinson's, and all of those different categorizations of patients would lead to different treatments. But I strongly believe that by looking at the data together and collectively, you will see subtypes in the data. You will see indices that are associated not just with treatment outcomes, but with differential outcomes that nobody had suspected. And I think that might be a challenge and an interesting area for many of us to look into now that we have all of this data in its first round of an analysis.
So the next question is around AI, perhaps more general than what you presented, but a bigger problem for all of us using AI. So AI sometimes makes false results, such as hallucinations. What can we do to use AI responsibly and consider these possibilities?
Yes, I really like that question. So if AI is used without being circumspect, a radiologist could use AI too. In fact, radiologists are very excited about AI. They can read in a scan. And their excitement about it was very different than I thought. If AI can pre-screen 1,000 scans that they have in their queue and bring one to the front that is acute, the AI doesn't have to make the decision. They can make the decision, but it can triage and identify things that they don't have the cognitive power to survey immediately. So I think using AI as an adjuvant, as an agent, I mean, all of us know this if you use AI in your everyday work, that is somewhat benign in the sense that the final decision-making has the radiologist in the loop. Now, I think to your point, Marta, if there are incorrect signs of disease that are flagged, and that leads people to an onerous or troublesome experience, I think a lot of oversight needs to be brought to bear on that. So many of the FDA requirements for an AI tool to be used in a radiologic context for diagnosis are to make sure it does no harm. So if there is a test that could be tremendously alarming, you don't want the AI lighting up all of the brain and saying that this is a problem. Mathematically, there's very interesting work trying to understand whether generative models that are at the core of many AI techniques are generating data or explanations that fit within the empirical range of data that has been used for training. So there are hallucination mitigation techniques. There are things that go in there and don't unquestionably accept the output, but go in and try and have secondary screens to make sure that these things don't get into the workflow in a harmful way. But what do you think? Are you excited about AI or modestly nervous about AI?
It's your talk, not mine.
I'm passing the buck. I don't know the answer.
I mean, I agree. I think it's a powerful tool, but we need to be able to use it responsibly. And I agree with what you said. At the end of the day, is the person taking responsibility ultimately in everything that we do.
So, Paul, we're getting quite a few requests that are very practical, very pragmatic regarding the nature of ENIGMA. And for instance, we have people asking how they could join the ENIGMA Consortium or a certain group. And more questions also regarding the access to the data. So we were just talking a moment ago about replicability, but also training AI models, maybe. So I guess we could rephrase the question by asking whether some of us could access the data from ENIGMA or a portion of it to train models, or maybe to just replicate some of the results or extend the results that your team has found.
Yeah, thanks, Sylvain. I think that's a great question. So many of you who've tried to get brain data from another lab might have had varying experiences. And in ENIGMA, we've tried to tackle this with several tiers of access. So if the question that you want to answer can be adequately answered by meta-analysis, the remote lab can analyze their data and send you the results. So with the workflows now being widely used across the world, someone can run VBM or they could run fMRI prep at another lab. This is the least level of data sharing. And in genetics, this is common. So in genetics, you tend to not ship whole genome data around for privacy reasons. But as we've seen, the ability to model individual level factors in data that might affect the results is not there with meta-analysis. You want the data. You might want derived data, which can be anonymized, and depending on your national law, you might be able to send derived anonymized data. Or you might come up with a methodological solution. Many of you are interested in federated learning. So having a client program that is operating on distributed neuroscientific data that doesn't ever leave the site where it was collected. But I do want to end on a practical note. If you want to do a project and you see something here, or maybe at OHBM in general, that interests you, we often ask the person who collected the data if they require a data use agreement, a bilateral agreement that is signed and specifies who will have access to the data, is the data secure, and whether the terms under which the patients provided the data are being respected. And the vast majority of cases, that works. I think, I would encourage you, if you're stuck with that, come and ask us about it. We'll try and brainstorm about how to get data. If you have a good idea, if you can think of a project you'd like to do here, maybe there's a working group on a disorder that you are expert in and isn't represented here, I think you'll find a very, very welcoming community. I mean, I think nearly all of the scientists are excited about the chance to learn from other people, and the data sharing will make sure it works. But I think that's a very, very big topic, and many of you wrestle with this daily, so I don't want to give a simplified answer. But I think depending on the degree of data that you would like transferred, you may actually prefer to leave it where it is, particularly if you're doing genetics.
So the next question is around RDoC. So the RDoC framework, going beyond diagnosis into more of a matrix type of characterization of mental health conditions. So how will ENIGMA work around that?
Thanks, Martha. That's a great question. So one criticism of ENIGMA, in fact, if you're wearing the ENIGMA t-shirt and here's a map of a disease, the RDoC people will say, and this was an initiative, if you're not familiar with it, that began at the NIMH, where they said people with schizophrenia, for example, might have negative symptoms, they might have positive symptoms. There are dimensions of their symptomatology that we shouldn't lump into a category. And you work on this, so you know this better than I do. So if you were to co-vary the imaging variation with a dimensional measure of each of the relevant symptoms, you might be able to have insight into what is going on in a much more refined way than if you lump people together. So in most of the grant applications, many of you write a lot of grant applications, the data is first analyzed categorically because as a matter of convenience, a person might study a disorder. But then very soon afterwards, the patients are not lumped together in a homogeneous category. I mean, we saw that, for example, there might be subtypes in the imaging and one subtype might be particularly locked onto a domain or dimension in the symptom data. And I think it's a second level of analysis that's actually quite tricky because you have to agree on a consensus as to how these symptom domains are evaluated and harmonized. But in the end might factorize the component elements of the disease in a more logical way, perhaps giving better insight into what is going on. Thank you.
Okay, so there is a question that relates to the bridge that you are building and indicating and showing the way through that bridge to AI and the applications of generative AI in particular in our field. And there are questions surrounding so you mentioned the interpretability of the AI models. This is still a challenge, especially in deep neural networks. But the question we have received from the crowd relates to dealing with uncertainty in the outcome of a certain classification, for instance, or certain generation of an image or synthetic data with an AI model. So how do we reconcile basically the tradition of our field, stemming from advanced statistics with what AI is about to offer to us?
Yeah, I really like that question. So Tom Nichols gave a talk at the last OHBM about conformal calibration, which is one answer to your question. So in radiology, people's appetite for AI solutions depends to some extent on whether the AI can report its confidence or degree of evidence for a decision. So if it were to read a scan and be honestly agnostic about what there is in the scan, many radiologists would be very, very happy with that answer because they say, well, I wasn't going to trust the AI anyway. It's just an adjunct. So I think one mathematical way, and many of you may have other thoughts and ideas on this question, is that if diagnostic classifiers or AI techniques that are trained to make a decision also report not just their best answer, but the confidence in each of a set of answers, that can be factored in. And the approaches that I'm learning about at this meeting, Tom Nichols has a very nice presentation on conformal calibration. They take some held-out data and try and assess the degree of accuracy in different contexts. And of course, if the imaging is poor quality, you'd like the AI to be honest and say, this isn't a decisive situation. But if it's absolutely clear-cut, the brain is full of amyloid, you would like it to say that is flagging amyloid positivity, for example. So thank you.
All right, in the next few minutes that we have left, Paul, so I guess we are all curious about two things. The first thing is the lessons you have learned after 10, 12, 15 years, growing ENIGMA and putting everyone behind you on this journey. Especially there are some very specific questions regarding, how to deal with the different jurisdiction in terms of data privacy, data sharing, ethics, consent, etc. But beyond these aspects, which are very important, what is your personal, lesson that you have learned?
It's such a good question. I think the most treasured thing that we each have is our network of collaborators and supporters. So you'll have seen a trend in funding towards big data for replicability, collecting more data. We need the data, that's part of it. But I think one of the lessons we learned, and many of you who collaborate with other labs know this, is that the diversity of perspectives, and I'll give you a concrete example. So Sophia Frangu is a prominent psychiatrist. Many of you know Sophia Frangu. And she said, I think the siblings of people with bipolar disorder have unusual brains. In a genetics class, you're told that people at genetic risk for a disorder have a phenotype that is on the way to disorder. And she said, I don't think that's true. I think these brains are very different, but I've seen it. My colleague noticed this as well. And all of these strands of evidence, I've been hearing them. She goes to a lot of psychiatry conferences. So she designed a study where she was looking at the siblings of bipolar patients, and they appear super normal. It's a very unusual result. And she said, I tried to get this published so many times. I tried to send this to journals. They thought it was ridiculous because the orthodox view of genetic risk is that people at high genetic risk have a phenotype that is intermediate between healthy and the disease. And this was the opposite. Now, she originated that study by asking a lot of people what they thought, comparing her observations with other people's observations. Now, you could say, did the data matter? The data did matter. If the other people hadn't collected the data, that would have mattered. But it was the insight and the community and the person's ability to aggregate and synthesize information that they were learning that led to a novel hypothesis. And I think this vision of the OHBM community is absolutely unique. It's a treasure that we all have. If you make connections with the other folks who are interested in related topics of the field, they'll guide you on a journey that you could never have imagined. I think you will all see this at this meeting. And so, although we tend to think of the barriers to be data privacy, data protection, many of you have solved this. Many of you have been able to interchange data to a maximum degree. It's a headache, but to some degree. But I think the main lesson, certainly the lesson we've learned, and we could ask other people involved, is that the diversity of perspectives and ideas is like gold. It's not zero-sum. You'll never run out of other people's ideas. You can just avail yourself of some advice, go ask them about what they're doing, go to their posters, go to their presentations. And it's like an infinite resource. So even though the data is finite, the creativity is not. And so to try and facilitate this, and we're learning, having junior people lead projects, having people from traditionally underrepresented communities lead projects. Emily Dennis with her ENIGMA U project, which is an educational project, bringing people in from countries that don't necessarily have access to neuroimaging education, and she's getting them on the ladder. I mean, she's doing a phenomenal job. So I don't know if you would agree, it's almost as though they're not technical aspects, but they're ones that we could take care of as a community that catalyze and create opportunities and lead us forward on a path that we might not have imagined.
And can you say a bit more about what the future holds for ENIGMA?
I think that's up to you. So if you have any good ideas of projects you would like to do, let's see if we can do them. AI, I mean, to give you a better answer, AI is paradoxical. So those of you who went to the mathematics and statistics workshop in the last 2 days will have seen this whole landscape of techniques that are emerging every day at lightning speed from all areas of computer vision, language, multimodal correlation. And I mean, I love this field. I would strongly encourage you to dip into it, engage with it, go to the workshops here that are on this. It seems like AI, as a branch of mathematics, has opened up a whole new landscape of discovery. But without your neuroscientific background, you will not be able to maximally use it. And so I think this hybrid area where neuroscientists who are now very conversant with brain disease, imaging modalities, and all of the technical aspects of brain imaging are going to be exceptionally well positioned to bring AI into our field. And who knows, I am excited about that. The flip side might be the modeling, and many of you who went to the workshops today who are looking at gradients or simulations or MEG, EEG, you know that the physics and the fundamental physiology of the brain is absolutely fundamental to what we do. So if some new mathematical technique comes along, it's nothing without an understanding of the physiology and the architecture of the brain, the systems, neuroscience. So I think we're very, very lucky. We live at a time where our trainees can learn the advances in mathematics and computer vision and natural language processing at a time when that is growing phenomenally. But we also have the ability to learn from each other in the neuroscientific field, and that will help us create models. You might create a model based on your knowledge and experience that the AI would have a great deal of difficulty finding. So it's an exciting time.
Thank you so much for this last answer, because I think it's time for us to thank you so much for this fantastic presentation and everything you do with ENIGMA.
Thank you very much.
Let's learn from each other over the next few days, indeed. That's a beautiful message, Paul. Thank you for that, too. Thank you, Marta, for being such a wonderful co-chair. Thank you all for posting all these questions. We are sorry we couldn't take all of them in the limited time we had. But again, let's learn from each other, and let's have fun tonight at 7:30. So let's say now. I'm not sure exactly where, But let's share a drink and a few bites. Thank you all.